I want to thank the conference organizers for inviting us to do this session. I also want to acknowledge the huge contribution that has been made to the HIV vaccine field by many scientists, clinicians, and the community. We have decided to structure this plenary to be complementary to both the HIV vaccine and cure field, trying to learn from each other to make more catalytic steps to get to the end game. So I want to ask the question, can a vaccine strategy contribute to both prevention and cure? And can we learn from previous efficacy studies, non-human primate research, and neutralizing antibody science to see whether we can make biological interventions that can both be used for prevention and therapeutic interventions? So what kind of immune responses are required for an effective vaccine? We need potent neutralizing antibodies, as seen from SARS-CoV-2 vaccines, the correlate between neutralizing antibody and protection. We need them to be broadly reactive. We need our immune responses to be persistent, and we need memory T cells that suppress viral replication. We also need immune responses that prevent viral escape. The rapid integration of HIV after acquisition and the inability for early therapy to interrupt this integration means that the goal of an HIV vaccine must be to prevent acquisition or eradicate recent infection. From studies coming from AMP and SARS-CoV-2, we see that it takes around between 50 and 100 times more neutralizing antibody to inhibit HIV than it does to inhibit SARS-CoV-2. So starting off with the correlates of immune response from prior infections, we want to see whether this will be a useful barometer to measure future vaccine strategies. So we've looked at the RV144 study, HV10505, HV10702, and HV705. And you can see from the Kaplan-Meier plots that the only vaccine that was moderately efficacious was the RV144 trial. So important correlates identified in RV144 provide us a lens to evaluate HV10505, 702, and 705. And for the purpose of this talk, I will focus mainly on the interplay between CD4 and CD8 T cell responses in relationship to both the breadth and magnitude of V12 antibody responses. So moving forward, looking at HV10505, which was a VRC DNA Ad5 heterologous prime boost, the correlates of protection were a combination of high CD8 T cell polyfunctionality and an antibody response which was associated with protection. The figure on your right shows the envelope-specific CD8 T cell polyfunctionality and its role in protection. And the table on your left shows the relationship between antibody responses and T cell responses and the role of it in protection. So very important, the first time we see a nice correlate between CD8 T cell polyfunctionality and protection. Looking at RV144, we see that the V1, V2 IgG was found to be an immune correlate of risk in RV144. This correlate has been confirmed in studies in different labs using different assays and reagents. The studies that have been expanded have included cross-clade antigens, AE sequences representing circulating strains, and a cross-reactivity score. So these studies, again, demonstrate that the breadth of the V1, V2 immune response correlate with HIV infection. Also, importantly, not only do we need breadth, but this slide shows the role of the levels or the magnitude of levels of IgG responses we need to induce protection. And this slide demonstrates the association between the magnitude or levels required for IgG to be useful in protection. This slide, this was a poster presentation at the IAS yesterday, and it looks at HV10702 and the immune correlates from a case control study, and again shows the relationship between high antibody V1, V2 responses in combination with a polyfunctional CD4 T cell response to achieve protection from HIV. And so here we see the interplay of high IgG responses together with a polyfunctional CD4 T cell response to achieve protection from HIV. Also, and as a late breaker yesterday, we had Ivy Kenny that presented some of the correlates of the 705 or Imbokoda study. And this slide I've taken from his talk shows again the breadth of the IgG3 V1 V2 response as a possible inverse correlate of risk in the study. So my message from the correlates of protection studies is that 3 neutralizing HIV vaccine efficacy studies, RV144, 702, and 705, indicate that high titers of V1, V2 antibodies may play a role in reducing HIV acquisition. We can't yet eliminate the V1, V2 hypothesis because neither 702 or HV10 705 induced high enough titers to confirm this correlate of protection as identified in RV144. IgG3 breadth was also a consistent correlate to decreased HIV risk in RV144, HV10 505, and 705. In HV10 505, the first efficacy trial that used the modality that elicited a significant prevalence of CD8 T-cell responses suggested that those with high CD8 T-cell responses after vaccination had reduced acquisition. So let's go on to non-human primate research. And I'm going to focus only on a particular non-human primate study, and that is with the rhesus CMV vaccine using the variant 681, which has some genomic rearrangements which may be particular to the interesting aspects of this vaccine. And I will show you both replicating and attenuated rhesus CMV studies using this vaccine. So the first slide I'll show you is around the replication or spread competent CMV vector using the 681 rhesus CMV. And this experiment was with four groups of monkeys looking at either 2 doses of the CMV or a dose of the CMV with an adeno compared to a DNA with an adeno and also as compared to controls. And here you can see in both Group A and Group B the reduction in viral load in macaques that had been exposed to the CMV vector. Again, looking at the attenuated rhesus macaque CMV, you can also see that in this study you get protection from this after CIV challenge, and this manifests as a rapid decline and ultimate clearance of the SIV from the macaques. So what is my message too? Is that it's possible that some CMV vectored we can use in human studies. In these 2 experiments, we saw that these monkeys were protected from SIV, and those who showed a rapid decline and ultimate clearance of SIV from the blood after acute viremia, which may be an important tool for functional cure. We also saw that the protected animals were also clear of reservoirs of RNA and integrated DNA, and that the protection was associated with antigen-specific CD8 T effector cells, recognizing epitopes presented by MHC class II and MAMO-E molecules. So we can see that the non-human primates can be a powerful tool and enable us to develop new modalities that can be advanced into humans. So moving on to the discussion around broadly neutralizing antibodies and vaccine strategies to induce neutralizing antibodies, I'll first start off with the AM study. This was a collaboration between the HP10 and the HVTN, and it occurred in 2 parts of the world, one in the Americas and one in sub-Saharan Africa, looking at different modes of sexual acquisition. Now, this study investigated a monoclonal antibody, VRC01, and showed that in people with sensitive virus, this VRC01 was protective. And this slide shows you the protected efficacy over time and looks at pooled VRC, low dose 10 milligrams per kilo VRC and 30 milligrams per kilo VRC. The blue line shows sensitive virus, and the orange and purple lines show intermediate and more resistant virus. And what you can see for those people who had sensitive virus, the protective efficacy was between 60 to 80%, and this was after 2 doses, and it was constant thereafter. But important, the protective efficacy to more resistant viruses was close to 0 at 80 weeks. Maybe we can move this neutralizing antibody or monoclonal antibodies forward, and one of the areas we're looking on in South Africa is whether there's a role of body neutralizing antibodies to prevent or eliminate breast milk transmission. So we know with breast milk transmission, there's a very narrow genetic bottleneck of founder viruses that causes breast milk transmission. And maybe we only need 1 or 2 BNABs to eliminate breast milk transmission. And so we are doing research in South Africa to see whether we need 1 or 2 and what are the best broadly neutralizing antibodies to move forward in the PMTC agenda. So what about whether broadly neutralizing antibodies have a role for antiviral control in HIV-infected people? And this study just looks at 2 powerful broadly neutralizing antibodies, BBCN117 and 1074, in people with HIV who were recently infected. And so after receiving the infusion of the 2 monoclonal antibodies, 3 days afterwards, there was a structured treatment interruption. And here you can see that those who received the broadly neutralizing antibodies remained off heart longer than those in the placebo, and also tended to have more controlled viremia post-infusion as compared to placebo. So the third message that I'll give is that AMP demonstrates that broadly neutralizing antibodies are capable of preventing HIV infection and provide the future roadmap for vaccine development. The target neutralization, which is more than 1 to 200, in the TZMBL assay may be a useful correlate in future trials. AMP has demonstrated also that we need to make broadly neutralizing antibodies to more than 1 site, and neutralizing antibodies may have a role in functional cure. Vaccine approaches need to shift to eliciting antibodies shown to known conformational structures that elicit such antibodies. So moving on to the last part of my talk, I'm going to talk about inducing HIV-specific neutralizing antibodies using active vaccination. And we've heard through the proceeds of this conference the approaches that are epitope-based. We look at lineage and germline targeting and look at stabilized trimers, and then look at different platforms like nanoparticles, viral vectors, and mRNA. And there are 3 major approaches to broadly neutralizing antibody-based vaccine design. There's the lineage-based that is led by Bart Haynes' group, the germline targeting approach, which is mostly led by Leo Stomatatis and Bill Sheaf, and then the immunofocusing approach, which is basically lineage and germline agnostic, and we'll see some more work happening in those areas. An interesting and exciting feature of these is the use of the mRNA technology to do this. And we've heard a little bit around the conference around mRNA as a strategy for an HIV vaccine. And this study in non-human primates provides an idea of how we can use mRNA to advance broadly neutralizing antibodies, and provides a proof of concept that we may be able to use various envelope options. Here you can see there was an autologous envelope without the 276 glycan, and then the glycan was reconfigured, and then it was boosted by an ANC heterologous boost, and then also introduced a protein. And in this experiment in non-human primates, you can see that there was a 79% per exposure risk reduction in those who had these series of immunogens given in an mRNA platform. Going forward, we anticipate using these kind of approaches, another 20 trials using an experimental medicine protocol approach. And already 9 have been reviewed and are in full protocol development, and we hope to move these along. And here you can see the various strategies and the various immunogens that we will be using in this experimental trials. Very important then is to iterate fast, and that's where the mRNA platform will become important. So my message 4 is that experimental medicine will set the pace to evaluate the concept of broadly neutralizing antibody vaccine design. And we are investigating this also in HIV-exposed infants in the HV10135 study that comes from Bart Haynes' immunogen. So the use of this new technology, such as mRNA, may allow us to rapidly iterate this approach of broadly neutralizing antibody vaccine design, but we need speed to advance this concept, and that's where the beauty of the mRNA platform might come in. So what about the end game? So I've showed you data from clinical trials in non-human primates, and has emphasized the importance and the critical role of neutralizing antibodies will play in protection from infection. We've also seen that we probably need cocktails of monoclonal antibodies for both for both prevention and a functional cure, and that mRNA platforms may be, of envelope proteins may be a useful tool to use to iterate to induce BNAb induction. But I'm going to say that we will need an HIV vaccine that requires a complex mix of immunogens, as we need more than one targeted approach. And we need to combine these antibody approaches to also make sure we use approaches that elicit significant cellular and innate responses, and this is important to pursue together. So there are a lot of people I need to acknowledge, and I'd like to acknowledge the funders, the people that there's been fidelity to making HIV vaccines. I want to acknowledge the enormous resources that have been put in to get us this far. And I want to acknowledge the HVTN that has done incredible work in moving us forward, particularly Larry, and I want to also thank Georgia and Troy for some insights on these slides. Also to thank Avi Violari from the HVTN 135 who enrolled the study in record, and the MRC and Mina as well, but also the study teams of 505, 702 and 705, and everybody involved in the HVTN, our colleagues at DAIDS, all the site staff, our global CAB, and our participants who participate at a global level in these clinical trials. Thank you very much.